All right, welcome to the course. In this course, we want to cover the molecular biology and chemistry basics you really need to understand to build models, nothing more. So, these days, we are designing molecules and delivery systems under defined constraints. That's why therapeutic development is now becoming an engineering problem. Second, we need to talk about the shift in the workflow from lab first screening to design driven pipelines where candidates are evaluated on computer before experiments. Third, how your existing engineering skills transfer. Concepts like modularity and feedback apply directly to biological systems. So we want to stress on this point. You know a lot more than you think you know about biology. And finally, where machine learning is making the biggest impact from structure prediction to data-driven molecular design. So let's start discussion with Nobel Prize. The 2024 chemistry Nobel Prize went for computational protein design and protein structure prediction. This is big. Molecules are now designed computationally before they are tested experimentally. Let's start with a little bit of history. In the past, pretty much of all the 1900s, drug discovery relied mostly on trial and error. It was slow, and obviously, you can guess that there was a lot of luck involved in discovering new molecules. Then things changed in the 1980s with the high throughput screening. HTS had completely industrialized this by the 2000s, but it was still mostly experimental, fundamentally a numbers game, testing kind of random candidates without prior design. And then we will finally see the true change. That is in the 2020s, you can think AI, ML first design. We have used computational models to support screening for decades, but the 2020s mark a specific tipping point. We have moved from just analyzing existing libraries to generating entirely new structures from scratch. With the tools like AlphaFold and generative AI, the computer is no longer just an assistant checking our work. It is becoming the primary engine for design. So what's different now is that we can design molecules on the computer first, predicting structure, binding, and drug-like properties before we even go into the lab for synthesizing. The workflow has shifted from test and see to design, then validate. And then we will see a new type of companies in this space. We are seeing a shift in who is building new therapies. Alongside of traditional pharma and biotech, a new group of tech bio companies is designing drugs using data-driven pipelines. If you look at these companies' websites or on LinkedIn pages, you would notice they are hiring software engineers, ML researchers, and data scientists because their work looks much more like modern data workflow than classic wet lab discovery. The core idea is simple. use large-scale data and models to guide molecular design. And then now let's see the architecture of biology and what you already know. We started saying that biology is becoming more and more, a data problem. Let's look at the system architecture. You will see that evolution actually arrived at many of same engineering ideas you already know. You can see some examples on this slide. We will see more in coming lectures. Let's start with feedback. Feedback loops keep systems stable. Homeostasis is simply a loop that holds values like temperature, pH within a narrow range. In your code, loops keep systems stable, right? So same idea. So on this slide, you can see a variable pH, but it can be temperature or something else. So the idea here is, it shouldn't be too low, too, too high and too low. So optimum level needs to be maintained. That's it. Next, let's discuss redundancy. Just like you have backups for genomes, backups for critical data, our genomes are deployed. This means we carry two copies of most genes so that single failures are often tolerated. And then let's see modularity. Nature also uses modularity. Organelles act just like subsystems or microservices. Each one handles a specific task like energy production or waste disposal independently of other functions, more or less. And then now let's see error correction. Just like ECC memory, DNA repair systems are constantly scanning genome and fixing mistakes to limit damage. Once you see these patterns we just discussed, the architecture starts to feel very familiar to you. And then we want to stress one thing, that is your skills already apply. Let's bring this all together. If you are coming from software, these parallels should feel natural to you. As you know, compiler, you can think that as a ribosome. Like compiler, it turns code into a program, right? So basically, ribosome is doing a similar thing, turns code into proteins. Think about database. That's like your genome. The long-term storage for instructions, even the cell, it behaves like a container running isolated process. You can see other examples here. Just one point. These are analogies, just for your conceptual understanding. Let's see why all this matters. So the key point is that therapeutic development is shifting from discovery to design. We are seeing entire companies built around these workflows where cloud labs and automation allow us to run thousands of experiments in parallel. And in this course, we are going to learn the foundations to build these models. Thank you.